So yeah, that's the, oh, there we go. So now we're recording. So I'm Constantine, and I'll be talking, like Gabriel said, about sort of my graduate research so far, and what I've done in terms of radiolabeling gold nanoparticles, creating radiation nanomedicine, and using it to treat glioblastoma in a preclinical mouse model. There we go. So we'll just quickly go over the outline of my presentation. I'll talk a little bit about glioblastoma and its treatment. I'll explain what is radiation nanomedicine. I'll go over the animal models and the methods that I used in my research, as well as the results for biodistribution, SPECT imaging, dosimetry, toxicity, and we'll finally look at therapeutic evaluation, as well as future directions for my research. So glioblastoma, or GBM, is the most aggressive and most common malignant brain tumor. And while rare compared to other types of cancers like breast and prostate, it is always fatal, and the median overall survival is only a little bit more than a year, 12 to 15 months, with a 5-year survival of less than 5%. So this is a very debilitating disease, despite standard of care treatment, which consists of surgical resection to the greatest possible extent, depending on the location of the tumor within the brain, external beam radiotherapy delivered in 2 great fractions to a total of 60 gray, with concurrent temozolomide chemotherapy given during the radiation treatment as well as afterwards. However, as I mentioned, despite this treatment, the overall survival and median survival remain very, very low. And these treatment challenges can sort of be summarized by talking about the residual disease that remains after treatment. So as I mentioned, surgical resection is sort of one of the first lines of therapy, where this neurosurgeons will cut out as much of the tumor as possible, but complete resection is impossible. You can get up to over 90% of tumor resection, but there's always a little bit remaining, especially due to the invasive nature of GBM, where you may have a bulk tumor here, but the tumor extends beyond that into the healthy tissue, and it's hard to resect that without removing large parts of healthy tissue. Additionally, the radiation is limited to 60 gray, and once there's reoccurrence, there isn't really a standard protocol afterwards. Additionally, temozolomide is the only chemotherapy used in official standard care treatment, and about 50% of patients with GBM have some sort of molecular resistance. It's the epigenetic factor that will reduce the alkylating damage that temozolomide causes to DNA. And then finally, as many of you probably know, the blood-brain barrier prevents the majority of chemotherapies from being effective. They don't cross the blood into the blood-brain barrier, and if they do, they're effluxed out by drug transporters. Established and novel drugs end up not making it into the brain, and there's no way for it to be effective if you can't reach the tumor in the brain. So like I mentioned, the treatment fails to eliminate residual disease. It's very good at removing the primary large bulk tumor, but there's always residual disease, and recurrence usually occurs 2 to 4 centimeters in the margins of the original tumor. And like I mentioned, there's no standard treatment for this recurrent disease. So what strategies are there? What strategies are available to solve this problem? One of them is a neurosurgical technique called Convection Enhanced Delivery. And like I mentioned, most therapeutic agents cannot reach effective concentrations in the brain after oral or IV administration. CED is a method where catheters are inserted into the brain, into the tumor region, and using an external infusion pump that creates a pressure gradient, a therapeutic agent can be infused into the tumor, bypassing the blood-brain barrier, and it is compatible with a wide range of therapeutic agents, including, but not limited to, proteins, antibodies, chemotherapy, polymer drugs, as well as various nanoparticles. And the way this is achieved is even these large molecular weight therapeutic agents, if this was relying just on diffusion, the diffusion of a large molecular weight compound is very slow. However, because of the external pump, we are creating a pressure gradient that means that the molecular weight is not a factor in terms of delivering this a therapeutic agent by CED. For my research, I focus on using gold nanoparticles, and I'll talk a little bit about why we selected gold nanoparticles and what we think they're good for. So to construct our radiation nanomedicine, we start with our gold nanoparticles. We have a radionuclide. I'll talk about a little bit about our selection later. as well as a chelator that sort of facilitates the connection of the radionuclide to the gold nanoparticle. And after a simple synthesis procedure, we end up with radio-labeled nanoparticles with a radionuclide strongly bound to the surface of the gold nanoparticles. And that ability, the ability to functionalize a gold nanoparticle is one of the reasons why we selected gold nanoparticles. Tying the radionuclide to a large nanoparticle alters the pharmacokinetics and the drug delivery of the therapeutic agent. And this is not unique to radionuclides. This is compatible with a large, wide variety of therapeutic molecules, like I mentioned, antibodies, proteins, drugs, etc. However, radionuclides are, in our opinion, are uniquely positioned for treating GBM because, for one, cancer has a reduced capability to repair DNA caused by the ionizing radiation. And also, the radiation will generate a predictable therapeutic field. So I made a little diagram here where, for example, there is a limited range to which the radiation will reach within the brain. Any cells within that range will be irradiated. However, anything outside will be safe. So this gives you a large degree of physical control of which parts of the brain can be irradiated, both first by the delivery of the drug being specific to the tumor region, as well as the physical property of the radionuclide. In order to accomplish this, our lab works with the Department of Chemistry here at U of T, with the lab of Dr. Mitch Winnick. Together, we've synthesized what we call a metal chelating polymer, which consists of three main sections. The first here is a PEG block that increases stability and reduces aggregation and MPS uptake. This is more of a concern for IV injection of these these radio-labeled gold nanoparticles. However, there are still cells within the brain that can take up and eliminate the nanoparticles, so this helps avoid that. The second part contains the chelator, which consists of a polyglutamine peptide with 8 pendant DOTA. So this is just the structure of DOTA. It's a very common chelator used in radiopharmaceuticals, and it strongly holds onto our radionuclide. And because there are 8 of them, it allows each polymer molecule to contain and hold on to a large amount of radiation. And the final part is, again, part of the polyglutamine backbone, but there are pendant lipoic acid groups where the sulfur atoms in the lipoic acid form a strong bond with the surface of the nanoparticle. Because there are a bunch of them, that connection is very strong, and it is unlikely for the polymer to dissociate from the gold nanoparticles. And then finally, from the radiation nanomedicine, you need the radiation part. So you can select a wide range of radionuclides. The 3 sort of main therapeutic particles that are emitted by radionuclides include beta particles, alpha particles, and OJ electrons. Our lab has experience with all of these. However, for my presentation, I'll be focusing on beta particles, which sort of represent a middle ground between the very short range OJ electrons and the higher energy alpha particles. These beta particles, also known, they're just electrons. They can travel multiple cell diameters, which is ideal for treating small to medium tumors. And additionally, the radioisotope or the radionuclide we selected, lutetium-177, is widely used in various radiopharmaceutical properties for different types of cancers. The production and the logistics of acquiring lutetium-177 is it's a very robust industry, so we can acquire large amounts of this with no issue. And so we get to sort of the hypothesis of my graduate research, which we believe that convection enhanced delivery of gold nanoparticles radio-labeled with lutetium-177 will be effective in controlling GBM recurrence. And it's just the sort of cartoon diagram of how we create that. And we believe that this intraoperative injection will be able to prolong the survival of patients eventually. But first, there needs to be a lot of preclinical work to sort of establish basis for that moving forward. So I'll get into sort of the animal models and the experiments that we can perform, and I'll talk a little bit about the preclinical model of glioblastoma that we use. So first, we inoculate our NRG strain mice with human GBM cells. The cell line is U251, and these cells have been transfected with luciferase, which will come back later. It enables us to perform bioluminescent imaging. Additionally, we selected NRG mice specifically because for one, they are immune deficient, like SCID mice. However, SCID mice have mutations that make them unable to repair DNA damage, which for radiation is a huge problem. These mice, the SCID mice, are extremely radiosensitive, and it makes evaluating a radiopharmaceutical product quite difficult, especially in the context of therapy. So these energy mice, like I said, they are immune deficient, but they don't have that mutation in their DNA repair pathway, so they can withstand more. The healthy tissue can withstand more of the radiation damage. From there, we wait for the tumor to establish for 2 weeks. After that, we go to the same stereotactic coordinates within the brain, and we inject 5 microliters of our gold nanoparticles or control injections, as you'll see later, depending on the experiment. From there, we can perform molecular imaging experiments, SPECT CT, I'll talk about MRI, as well as BLI, like I mentioned earlier. We can perform biodistribution, by which we collect the organs from the mice at different time points and measure how much radiation is in each organ at each time point. We can evaluate toxicity in terms of toxicity to healthy organs. And finally, we'll look at the therapy results. I'll start with our SPECT, or single photon emission computed tomography imaging. So this is a technique by which if you inject a radionuclide that emits gamma photons, lutetium is an example of one of those isotopes, which means that we can basically, using a SPECT system, it's basically, some people call it a gamma camera. It's basically a camera that can detect gamma photons. So if there is radiation in the mouse, we can image it and find out where it is in the body. This is just an example of a bone scan. So you can see a radiopharmaceutical product was given to this mouse. The product, or the drug, was uptaken by the bones, and then you can see the skeleton of the mouse from the SPECT image. For our purposes, however, we looked at just the head of the mouse, and we injected 1 megabecquerel of our lutetium labeled gold nanoparticles into the brain. You can see here this hot area in the center of the brain is where the nanoparticles ended up. And as you can see, from 0 all the way up to 21 days, 3 weeks after injection, there still remained a large amount of radiation where we injected it. And just as a caveat, this is not decay corrected, which means that you're seeing here is the decay of the physical isotope over time. If you contrast this with an injection of just the polymer with our lutetium, the scale is much different. It's only 3 days, but by those 3 days, there is very little remaining in the brain. This sort of I was very lucky. This is one of the first results that we saw when we were doing these studies. But it basically gave us, it backed up our rationale for using the gold nanoparticles, as they can act as sort of like an anchor for the polymer, allowing the radiation to stay in the brain for long periods of time without redistributing outside of the brain. As you can see here, the polymer is either degraded or it's small enough to be redistributed outside of the brain. So this is a really early but really important result, showing sort of validating our rationale for using the gold nanoparticles. Another way to look at this was to measure the whole body retention of the radiation. It's a very crude experiment. We take our mice after injection, we put them in a falcon tube, and then using a dose calibrator, which is just a way to measure the radioactivity of a sample, we can measure how much radiation is remaining in the mouse at different time points. And you end up with something that sort of resembles a pharmacokinetic elimination curve. So here you can see in red is the gold nanoparticles, blue is the polymer without the gold nanoparticles, and matching up sort of with what the SPECT showed, there was a precipitous drop-off of the radiation from the whole body. So this is not only was the polymer redistributing away from the brain, it was being eliminated out of the body entirely. So by 5 days, there was basically no radiation left in the mouse. However, with the gold nanoparticles and that anchoring effect, it was able to retain about 75% of the injected dose for about 2 weeks after injection. And this is decay corrected, so this is not counting physical decay. Further sort of evaluating the biodistribution and sort of looking at where the radiation nanomedicine goes after injection. We perform biodistribution studies. This is what I mentioned, where we sacrifice the mice at certain time points, collect the organs, and using a gamma counter, we can measure the amount of radiation in each organ. This allows us, if you do this over several time points, you can measure the cumulative radioactivity in the critical organs. We can obtain these S values from published literature, and we can use this, we can put this all together using the MERD equation, which is the medical internal radiation dose equation shown here, where in simple terms, you end up looking at the area under the curve over time for each organ. And we can calculate how much radiation dose is deposited in each organ by the other organs in the body. This is sort of one of the models and what it looks like here. You can see there are different organs. You can see like sort of the spine, the lungs, et cetera, the brain. However, because our injections were directly into the brain, we created a sort of unique model for the brain, where here it's a very simple diagram, but we modeled basically the left hemisphere of the brain, the right hemisphere of the brain, the cerebellum here, as well as the tumor volume as well. So we subdivided the brain in order to get a better idea of how much radiation dose was delivered, not only to the tumor, but to the healthy portions of the brain as well. And here you can see these are the results from biodistribution from 1 to 1 hour, 24 hour, 72 hour, 1 week, and 2 week after injection. And here's just another model to remind you of how we divided the brain. The gold here represents where the injection site was and the tumor. And as you can see here, this, just to draw attention, this is a split axis here. For most organs at most time points, the amount of radiation found in those organs were very low, and were low for the entire duration of the injection. So from here, again, you can see much less than even 1% in most of the organs outside of the brain. And very importantly, if you look at the different components of the brain, the right hemisphere, the left hemisphere, and the cerebellum, there is even very little redistribution of the radiation nanomedicine from the injection site. So very little was crossing over into the left hemisphere or escaping sort of to the back into the cerebellum, which is great. It's what you want to see from these injections. It's it's controlled physically directly where we wanted it, and it's stayed there over time. From there, using all these values, we can, with the help of Dr. Zhang Kai in our lab, we calculated the radiation dose estimate from these injections. And it makes sense that if most organs weren't exposed to radiation, there's very little radiation absorbed dose over time. So you can see here, much less than even 0.1 or 0.25 in most cases here. Even the whole brain, where you combined all three of those components, the absorbed dose was much less than even what is delivered by external beam radiotherapy. Additionally, like I said, the left hemisphere, the non-tumor bearing side, very little absorbed dose. The right hemisphere, which we modeled as the healthy tissue surrounding the tumor, the sort of the closest proximity to the radiation, only received about 6 gray, which is very little, especially for a fairly significant portion of the brain. However, if you look just at the tumor, sorry, I should click this so you can see the box, but the tumor itself, we estimated about 600 gray was delivered directly to the tumor, which is about 10 times more than what is delivered clinically. So really great results that sort of, again, are validating our hypothesis that the injections stay in the brain. They are not damaging to other organs as well. And just to further confirm that there's no other toxicity, we looked at from blood measurements at 2 weeks after injection. We looked at a panel of different biomarkers, including alanine aminotransferase, creatinine, glucose, total protein, white blood cell, red blood cell, hemoglobin, platelets, alkaline phosphatase, blood urine nitrogen, and hematocrit. And this was compared against control mice with compared to a 1.5 megabecquerel injection of the lutetium-177 gold nanoparticles. And as you can see, there are no difference in any of the values, which is great. That's what you wanted to see. And then just to confirm body weight as well in this sort of very acute time point, while there is some very slight dip in the average weight, it's not statistically significant. And later, when we get to the therapy results, we'll see that this small dip is not, it doesn't continue for the entire study. The mice treated with the lutetium nanoparticles actually increase in weight. We'll get to that in a little bit. But before we do that, we're gonna have an audience poll. I think Gabriel's gonna take over and run a poll just quickly in the middle of the presentation.
Thanks, Constantine, for the great discussion so far. As Constantine mentioned, we would like to ask a couple quick poll questions, and the questions are the following. The first question is, do you use or are interested in any of the following modalities in your research? The options are MRI, optical imaging, PET-CT, ultrasound imaging, photoacoustic imaging, DEXA, and intravital microscopy. The second question is, which of the following would you benefit from implementing in your lab? The options are MRI, optical imaging, PET-CT, ultrasound imaging, photoacoustic imaging, DEXA, intravital microscopy, and please select the most appropriate response. We'll just wait a few moments to let everyone answer the questions, and after that, we'll continue with the presentation. We'll just wait a few moments more. Okay, perfect. Thank you for answering the poll questions. I will now hand over the presentation back to Constantine.
Great, thanks. So we'll now get into sort of the therapeutic evaluation of the injection of the radiation nanomedicine. And we'll start just in ways, different ways to monitor tumor growth. And as some of you may know, bioluminescence imaging is a great non-invasive way of measuring tumors in vivo, especially for GBM, especially because, you know, if you have a flank tumor model, you can obviously measure the tumor volume just with calipers or something and calculate a tumor volume based on that. You can't do that for inside the brain. So bioluminescent imaging is a great way to sort of get a correlated measurement of tumor volume or tumor size. Basically, the cells and a bunch of other organisms can be transfected with firefly luciferase. When you inject the mice with luciferin, the substrate for that enzyme, those cells take up the luciferin. The reaction of the enzyme ends up producing light, and those photons can be acquired using specialized cameras, and we can end up with a way of quantifying tumor growth over time. As I mentioned, these U251 human GBM cells were transfected with firefly luciferase. And the second way I measured tumor growth was by T2 weighted MRI. So just a very, very brief explanation on how T2 weighted MRI works as opposed to T1 weighted. T2 weighted MRI, a magnetic field basically takes all the protons in the water in the brain or any other part of the body that you're measuring, aligns them all. And then as the magnet turns off, they all end up randomly spinning back and relaxing into a sort of a random orientation. And you can measure this using MRI systems and it'll generate an image. And that's basically how the T2 weighted images work. You don't need contrast agents. That sort of works with T1 imaging. You can still get great images of the brain. And we used the M3 aspect 1 Tesla system, which is low powered compared to 7 Tesla systems, but was still able to get really good images, as you'll see in a couple slides. And I think the best part was that I could do it all myself. You don't need a team of experts to run the MRI. You can do it. Even a grad student can learn how to do it. So we'll go from the BLI here. And just on the left here, you can see a tumor growth index, basically compared to day 0 after from treatment. How did the tumor grow? The saline and the red in blue, the red is the non-radioactive nanoparticles that we injected. So just the gold with the polymer, no radiation at all, just to see if the gold nanoparticles themselves cause any therapeutic effect. And from BLI, you can see there's no difference in the exponential growth here. However, for only 1 megabecquerel of the lutetium labeled gold nanoparticles, it basically completely eliminated tumor growth over the first 3 weeks. And here you can see just some representative images from the treatment groups where the signal here is just, it's the computer colors it in. So you can visualize how much signal is coming out of the brain. And you can see here, just this one mouse in this one group had a little bit of signal left over. The other mice did not, which is great. A really good, very fast way of measuring tumor growth over time. And then as I mentioned, the MRI. So here you can see those same groups, the saline control, the non-radioactive gold nanoparticles, and the treated group. These are just, again, representative images taken on the MRI. You can see here in the front right hemisphere of the brain where the tumor was inoculated, you could see a massive tumor forming after 4 weeks after treatment in the saline control group. A similarly large tumor here from the non-radioactive nanoparticles. However, the treated mice with the lutetium-177, the tumor looked completely normal. There was no tumor that was able to be found on MRI. And we performed this on all of our mice, calculated a tumor volume, and you can see here, the saline group had the largest tumors. The radioactive, non-radioactive nanoparticles surprisingly had some mice with a small tumor. We'll get to survival in a second and show that this ended up not correlating with survival at all. But the 1 megabecquerel lutetium gold nanoparticle injected mice had basically no tumor across every single mouse that we saw. Here, just underneath our MRI images are when the mice reached endpoint, we collected the brains and sent them for H&E staining. And I'll be the first to admit these slices are not the best. You'll definitely find better histology slides out there, but it gets the point across where you can see here at endpoint the tumors are quite large in both of these groups. However, this mouse at the end of our study, there was no residual tumor left there. However, there wasn't a large tumor visible. If you zoomed in, you could see some residual tumor tissue remaining on the stain here. That's on the left in the sort of darker purple. And the tumor margin here looked completely healthy as well based on this H&E stain. We couldn't observe any sort of radiation, like necrosis or radiation damage based on the radiation treatment. And then very importantly, if you looked at the contralateral, the left hemisphere, so the side that didn't get irradiated of the brain, all this tissue looks completely normal as well. So again, just sort of using the histology to back up some of the toxicity measurements that we took earlier. Those blood measurements were sort of like a whole body, like holistic toxicity to see if anything was happening there. This is sort of like a local way to look at toxicity, and these injections again looked completely fine, which is great. And then in terms of long-term survival, even though, like I mentioned, sorry, I'll go back a couple slides, this non-radioactive treatment group had some mice with lower tumor volume. It did not impact survival. So you can see here that at about between 40 to 50 days, all of the control group mice required euthanasia because of the tumor growth. However, the lutetium-treated group, while there were mice that did end up having recurrence, their survival was pushed more than double compared to the saline and non-radioactive gold nanoparticle treatment groups, which is fantastic. And even more surprising than that was that 5 out of 8, so even more than 60% of the mice by 150 days were still healthy, and by the time we ended the study. So really fantastic results, very surprising. I don't think, I certainly didn't expect this, this ability to cure some of the mice, at least over the time period we looked at. We were expecting some improvement in survival, but maybe, some of the mice, they all end up recurring. We just pushed survival, but some of them ended up, at least from the study period, they ended up effectively being cured. And as I mentioned, we also tracked body weight over time, and you can see here, the body weight increases, and then as the mice get sicker and sicker, their body weight, is anyone who does animal work knows, the sicker the mice, the lower their body weight. You can see the drop off there. And the treated group, however, maintained high increasing body weight over the length of the study as well. So again, fantastic results, and I'm really happy with this working out the way we sort of anticipated it might work. But the success, you never know, is never guaranteed. So great results. And sort of just to go over a summary, and then I'll move on to sort of future directions as well. Stoma, like I mentioned earlier, it remains very difficult to effectively treat, but convection enhanced delivery represents a fairly new neurosurgical technique that can be used to bypass the blood-brain barrier and enable delivery of therapeutic agents directly to the brain, especially, again, in our opinion, radio-labeled gold nanoparticles could be uniquely suited for this task compared to organic nanoparticles that might break down very quickly over time, stuff like that. We can use SPECT and biodistribution to sort of track the gold nanoparticles in vivo. And then as we saw, local delivery was confined to the tumor with a large radiation absorbed dose, only to the tumor, very little systemic exposure, uneven exposure within the brain. We did not see any toxicity from the lutetium gold nanoparticles, and MRI or molecular imaging, MRI and BLI are powerful tools that we use to evaluate tumor growth. And very fortunately for us, this treatment was very effective in controlling tumor growth. So I'll quickly just go over some of the future directions of my work. Sort of this, all of these results were a great sort of foundation, but the model is fairly simplistic, given that they're not immune, they're immune deficient mice. We're not comparing against other treatments. This was sort of a great proof of concept, and again, really promising for expanding on this concept with more complexity. So, like I mentioned, one of the future directions that we're taking this project is to look at combination with checkpoint immunotherapy. And as many of you may know, if you're a cancer researcher, immune system involvement in cancer and sort of harnessing that potential is very hot right now. This is sort of generally how the immune system can be harnessed and used to end up treating the cancer. So the tumor ends up shedding antigens, so fragments of DNA, proteins, stuff like that, that your immune cells can pick up, present to T cells. T cells can be activated, see these signals as foreign, travel to the site of the tumor, and end up mediating some tumor cell death from this. But this is how it would work normally. Tumor cells, as many of us know, are quite smart and great at evading the immune system. And one of the ways they can do that is by expressing this protein PDL1, programmed cell death ligand 1. When the T cell finds the tumor cell, this sort of handshake happens where PD1, the protein on the T cell, sort of does like a handshake with PDL1. And this results in deactivation of the T cell and then no more cell death. So this is one way for the tumor cell to evade the immune system. However, with the development of immune checkpoint inhibitors, antibodies that bind to various cell surface receptors, you can sort of block this handshake from happening, where an anti-PD1 antibody will bind to PD1, for example, on the T cell. This handshake no longer happens, and the T cell can still launch an immune attack and result in tumor cell death. This concept works really well with radiation because radiation here, and I'll just focus on this sort of part of the panel, where this is just a slide from a review article looking at thoracic tumors. But the same concept can be applied to glioblastoma, where the primary tumor, the only tumor in GBM's case, receives the radiation. There is cell death and necrosis. This activates the immune system more than the tumor would normally, right? You end up having cell debris, danger signals, DAMPs, cytokines, lots of stuff for the immune system to pick up on and activate the immune system. So, like I said earlier, the APCs will pick this up, go to the T cell, activate them, but if you have a blocked interaction, it's useless. So combining the radiation with anti-PD1 therapy, there are other checkpoint inhibitors as well, but PD1 is one that shows that could be potentially useful for GBM. You can sort of work these two things together and synergize and potentially have a result that is greater than the sum of the parts. That's sort of the hypothesis between synergy in terms of like cancer treatment. And one of the ways you can sort of evaluate this in vivo is you treat the mice with your checkpoint inhibitor and the in our case, the radio-labeled gold nanoparticles. You can take the whole brains from the mice. You can do a really complex all-day procedure to isolate some of the tumor infiltrating immune cells. So those T cells will travel to the brain and hopefully help in killing the cancer cells. And then using FACS or flow cytometry, you can stain and sort these immune cells and calculate how much more activation there is of the immune system after different treatments. And this is one of the results I've been working on. They're not ready to share, but hopefully by the end of the year, we'll have another paper out looking at the immune checkpoint combination with our radiation nanomedicine. And before I close, there's just a couple of people I'd like to acknowledge for help conducting this research. So first, my supervisor, Dr. Raymond Riley, for his support over the past couple of years of my PhD work, my committee members, and then special thanks to STAR, where we conduct all of our molecular imaging and radiation work in vivo. Tisha and Deborah over there have been essential in, we transfer our radiation over, they have to sign for a bunch of papers, they help us with the imaging and stuff like that. And so none of those results happen without them. Actually, some help from Syntica as well. Tanya reached out to me sort of in 2020 when I was doing the MRI stuff. We were having some trouble getting really clear images, and she helped us put together a protocol, an MRI protocol, which was fantastic because it saved us a lot of time in trial and error and stuff like that. So she was a really big help. And like I mentioned earlier, Dr. Zhongli Kai, who's here in the top right, none of this gets done without her because she's by my side the entire time helping me with, she helped more with the dosimetry calculations. She's a dosimetry expert, but she's also helping me with doing all the experiments and paperwork and all that other stuff that you need to have for a grad student to actually have some good results. And I'd also like to thank all my funding agencies down here. And I think it's about time for a Q&A session. So if you have any questions, enter them, I guess, in the chat. Gabriel will help facilitate all of that stuff. So, and thank you for your attention.
Yeah, thanks, Constantine, for the great presentation. As Constantine mentioned, we're now going to move on to the Q&A section, with the first question actually being, what is the usual size of a recurrent GBM? Wouldn't the mean tissue path of Lu-177 cause a necessary dose to nearby health tissues if we are only talking about small lesions? Also, Constantine, if you don't understand when I pronounce
that's okay.
a certain word, yeah, okay, yeah.
I got it. Yeah. So I assume the question is talking about clinical stuff. And one thing I didn't mention in detail was the path length of the beta particles emitted by the lutetium 177. The maximum range is only 2 millimeters. So if you're talking clinically, 2 millimeters is a lot. It's very, very, very small for a human-sized brain, right? So if you can inject these nanoparticles along a tumor margin where there's some proportion of tumor cells, you will not end up irradiating very far past those 2 millimeters. The average range is actually 10-fold less, so it's 0.2 millimeters. So you're really focusing on a very, very small portion, a very small treatment volume. There are other radioisotopes that have much longer range. For example, just off the top of my head, yttrium-90 is one. That has a maximum range of a little bit, I think if I'm remembering correctly, a little bit over at 1 centimeter. So I guess the question is sort of, lutetium is sort of, like I mentioned, one of those ideal isotopes with the physical properties that enable you to sort of very, very specifically and physically control where the dose is being deposited. I hope that, I think, answered the question, but thank you for that question. It's a good one.
Awesome. Great for answering the question. Following to the previous question, here's the next one. Are there any previous stability studies done on the 177 LU CED that you used?
For the CED, no. To our knowledge, this is the first time gold nanoparticles have been used for CED. However, one of the things I didn't talk about was all the previous work done with this polymer in the gold nanoparticles. I think I briefly mentioned that it was used initially IV, intravenously, for different tumor models by previous students from our lab. So when I came in, a lot of the development of that nanoparticle, the stability testing, both of the lutetium to the polymer itself, and the polymer connection to the gold nanoparticle. That was all previously performed and published. You can, if you Google the Riley lab, some of the papers will come up from Simyoung Yuk, I think is probably the primary author on most of those. So a lot of that work was done by previous students, and I was very lucky to be able to go straight in vivo and sort of build off of what the work of the previous students there. But yeah, we've shown that it's very stable and doesn't separate, basically.
Great, thanks for answering. The next question is, why do you hypothesize the tumors were smaller in the non-radio labeled gold particle group?
Yeah, I'll just go back to that slide. It was a little bit of a weird result because, like, as you can see here, like there's a couple of them that have pretty small volume. Gold nanoparticles, depending on what papers you read, can have some therapeutic effect in terms of they can generate like reactive oxygen species and stuff like that. And that could be one of the reasons why the tumor volume was lower. However, I mean, the signal, the on average was a little bit lower and the standard deviation was a little bit tighter. So there might have been some early effect in the nanoparticles like that. We can't know for sure. That's one of the reasons hypothesized. But like I mentioned, in the survival here, there is like on average, like the mice live a little bit longer, but again, not statistically significant. So I think any effect of the non-radioactive nanoparticles, it's small and probably pretty hard to tell the difference. There are some papers that have injected intravenously a lot more gold nanoparticles and used them in combination with external beam radiotherapy. I think like the Hainfield papers were like some of the first to show this. So you can work with a non-radio labeled nanoparticles and end up having some control in tumor volume, but yeah, it's hard to tell based on, I mean, it's just the MRI images and sort of, we were sort of working with what we dealt with and those are the results. But yeah, we don't think it was anything sort of like meaningful, even though there was some measurement differences that we could detect. The survival ended up being basically the same.
Awesome, great. Thank you for the response. Next question is, well, it doesn't start with a question, but more like, thanks for the interesting presentation. And the question is, did you have any collateral effects on mouse related to the orthotopic implantation and injections?
I'll try to interpret collateral, but these methods, I don't know if the person wants to like clarify what they mean by that in case I'm misinterpreting. But in terms of collateral, the injection is the surgical procedure to do this injection is fairly, I mean, it's obviously invasive, you're injecting into the brain, but in terms of like work you're doing on the mouse, it's quite small. I'll just briefly go over sort of what the procedure is. I'll just leave this slide up. Yeah, it doesn't even show. Okay, anyway. But basically you end up cutting a small slice of the scalp away, or you make an incision in the scalp, open up the skull, you drill a small hole into the skull, and then that's where you insert the needle. This is something that's been done, I don't know, probably decades at this point in terms of establishing GBM models in mice, and then also doing the intracranial injections. So the mice all recover from the surgery when it goes well, and there's no sort of effect of the treatment of the surgery on the mice, if I'm understanding your question correctly.
Okay, thanks for your response. Next question is, how do you know the limited survival of the non-labeled group was due to tumor growth as opposed to gold nanoparticle toxicity? Did you perform non-radio labeled controls without tumor?
So for the tox, sorry, could you just repeat that last part?
Sure, yeah. Did you perform non-radio labeled controls without tumor to assess toxicity of the gold nano?
Without tumor, yeah. Okay. Yeah. So we didn't. However, we didn't measure the toxicity that I showed. Here in this panel, we weren't anticipating any toxicity from the gold nanoparticles themselves, and as you can see, their survival wasn't impacted compared to control. In terms of non-tumor bearing mice, we didn't. Well, actually, I'm trying to remember because this was a while ago. I think we did try and see if there was any toxicity from the gold nanoparticles in non-tumor bearing mice, and we found that there wasn't. Obviously, I didn't show that here, but as far as we know, the gold nanoparticles themselves aren't toxic and aren't causing the lethality. It's related to the tumor, especially because when we looked at these treated mice with the tumor, their symptoms were resultant of a tumor growth. You could see stuff like doming. You could see like paralysis, for example, in some of the mice. Those are sort of the reasons. Those are sort of the symptoms of large tumor growth in the brain rather than the nanoparticles. So we're pretty confident that the nanoparticles aren't toxic themselves, even though they might have some cytotoxic effect on the tumor. But we didn't evaluate that for certain.
Awesome. Thanks for the response. The next question is, do you think there are any other interesting radionuclides that will be useful for your work?
Yeah, for sure. I mean, I mentioned I'm going to try to go really far into the beginning of my presentation. But alpha particles are super exciting right now in terms of in the radiopharmaceutical field alone. I think maybe like 5-6 years ago by now, there was a paper published showing a patient in Germany, I think, had probably 100 or more prostate cancer metastases. And only after a couple cycles of an actinium labeled radiopharmaceutical product, one for PSMA, he was basically cured of the hundreds of metastases that he had on his body, which was like really incredible. And I think really like took up what people thought about alpha particles and showed that they can be really useful due to their physical properties. They do have a pretty small range and they're very high energy. So there are a lot more, there's a lot more potential for toxicity from that. But I think if we could swap out, it's also chelated by DOTA. So I think that using actinium could be really useful if another student wants to try that. There's also another radioisotope called, I believe, terbium-161. It has almost entirely the same properties as lutetium, the same like half-life, the same like beta particle energy, but it also emits Auger electrons, which are sort of very, very short range, but very densely ionizing electrons as well, compared to the beta particles, which are much less densely ionizing, but they travel further. So I think that would be a really interesting radioisotope to study. I think one of the problems is that it's not produced nearly as widely as lutetium is. I mean, like lutetium, there are like significant industrial processes and radiochemical processes set up to create large amounts of lutetium. I don't think that's the same way for terbium, and even actinium. There's it's a very high in demand, and the production of it is basically, I think there's like 2 or 3 places in the world that can even extract it. And you extract it, you don't produce it like you do lutetium, like in a nuclear reactor. You extract it from nuclear waste. So there's a lot less of it, even though it's very interesting and potentially very powerful for radiopharmaceutical treatment. So yeah, I think those are 2 off the top of my head that are really interesting.
Great, that was a great response. The next question is, why do the biodistribution results for the R hemisphere go above 100% ID per gram?
Oh, okay, yeah. So, this is injected dose per gram, and this is it's like a mathematical calculation where you compare the amount of radioactivity injected compared to the radioactivity in the organ at different times, and you divide it by the weight. So when it comes to these right hemisphere of the brains, they're pretty small weight, so you end up dividing by a number less than 1. So you end up having a larger percent ID per gram, and that's common for work like this, where you have a lot of radiation in a really small point, a really small mass of tissue. So yeah, it's just sort of a mathematical, not artifact, but consequence of how we calculate percent ID per gram. If you remove the per gram part, I think it would all look more normal. But yeah, this is just how it's calculated.
Yeah, thanks for the response. We have time for one more question, and it's the following. What are the further steps that are needed for clinical use?
Yeah, so I mean, I think these are all really good first results. I'm just going to go back to the Q&A slide. Yeah, these are all good primary results. Obviously, I think this needs to be, if this is to be translated and pushed towards clinical stuff, doing this in a larger animal model, so something like maybe like a pig, would be interested, and then having like a neurosurgical team evaluate this in a pig, doing sort of maybe similar experiments. You can do imaging on the pig as well to see if the results translate one-to-one. There are neurosurgical teams that do this technique clinically right now. It's not standard across the board. There's a group, I think, in New York who has done this with radio-labeled antibodies rather than nanoparticles. So I think it's definitely possible. I think it just needs this is just a lot of groundwork. There's just a lot more development that needs to be, evaluation that needs to be done in more animal models that are closer to human. And it would also be nice, our lab works with radio-labeled antibodies as well. And some of the projects have led to a little, like a kit development where it's basically like a vial of the antibody that you add your radiation, you shake it up for a bit, and it's ready to go. I think if we could develop something like that for the nanoparticles, I think that'd be really cool. It'd be a little bit more complicated than just like adding the radiation, shaking it up, and then you're good to go. I think it would have to be a couple more steps in that, but sort of making the production side higher volume would also obviously be, I think, necessary and potentially really interesting for future research. So yeah, another good question.
Great, thanks. So we've reached the end of our session for today, and to be respectful of everyone's time, we're just gonna wrap things up. As mentioned at the start of our webinar, we will be sure to answer any questions in the written transcript, and we will work to get this out to you over the next few days. I would like to thank Konstantin for the great presentation, and I trust that we have been able to provide you with a better understanding of evaluating intracerebral injections of radiation nanomedicine in a preclinical mouse model of glioblastoma. If in the coming days and weeks you have more questions about the modalities discussed today, I encourage you to contact us here at Syntica, and we'll be more than happy to discuss them further. Thanks again to all of you for taking the time of your day to attend our session, and we look forward to seeing you at a future Syntica event. Have a great day.
Thank you, everyone.
Thanks, Konstantin.